Hey, good morning, seventh graders. It's me, Mr. Stark. Today it is Monday, and last week we ran through the Microscope Lab where you had to look at the letter E at some of your cheek cells and the onion under varying different magnifications of using the microscope. So I just wanted to, I put together a little slideshow to show you what you should have seen, I was hoping that you would be looking at. So the first thing was you had to take a newspaper or a magazine and cut out the letter E and then prepare a slide with it by putting the slide down, putting a drop of water, placing the letter E on it, and dropping a cover slip on it at a 45-degree angle. This is one that I cut out of the magazine. It was the letters T, H, and E, and they were pretty small, so I just left them together. It was easier to manipulate and to put on the slide, and it also helps you with the orientation of the letters. So when I put the slide on, held it in my hand, this is the orientation of the T, the H, and the E. Okay, so when we started and looking under the microscope, through the eyepiece and the 4-power microscope lens, objective lens, which we call the scanning lens, this is what we saw. We saw the T, the H, and the E, and we could see that it was kind of flipped around and inverted. You could also see that the ink, that the letters in the magazine were not perfect. They were fuzzy on the edge, and then around it you could see the red, the blue, and the yellow little dots of the ink from the printing process. Okay, and then after we looked at that and we kind of were asked to play around with the letters a little bit and move them around and see what happens. So when we moved the slide to the left and was looking through the microscope, the letter E actually moved to the right. And if we moved it to the left, the letter E would move to, if we moved it to the right, it would move to the left. If we moved it up, the E moved down. And if we moved it down, then the E moved up. So everything was just reversed. So that's something to be aware of with the microscope, is that things get flipped around. And we also saw this with the hand lens, the magnifying lens over against the wall. Okay, and the next thing that we did was to look at them not only under 4-power but to 10 power, the 10x up here. So 10 and 10 on the eyepiece gave us 100 magnification. We can still see even more fuzzy and more of the red and blue and yellow dots magnified. And then the analysis here. As the E appears, they ask you what happens to the E. It appears upside down and reversed through the microscope. The ink appeared fuzzy on the edges of the black, and you could see red, blue, and yellow ink dots that were also visible. And the specimen must be thin to allow the light process of it to pass through. You would not be able to see the specimen if it were not thin. Okay. So if we didn't have a very thin piece of paper, and it wouldn't allow the light through, it would be very difficult for you to see. So if you had thicker paper, it would not work out so well. After the letter E, you were to look at a cheek cell. Take a toothpick and scrape the inside of your cheek, almost to the point of it hurting, and then smear those cells, that saliva and those cells, on a slide. Okay, lots of people asked about this black line right here. This is a pointer. These other lines are scratches in the slide itself. There's kind of a yellow hue or an orange hue to this because we took those cheek cells and we stained them with iodine so that they would show up better. The other thing that you may or may not know is that I used my cell phone to take all of these pictures, like this one right here of the letter E. I just shot down the tube of the body tube of the microscope, Same here, same as here. And this is under the 100 power. So the cheek cells are pretty small. And then under 400, we magnified them even more. This line right here is actually a scratch in the slide. And lots of the skin cells or the cheek cells from inside your mouth got caught up and hooked up on this scratch in the thing, so it was easy to find them. These dots are probably air bubbles, and that's the pointer again. So this is under the magnification of 400. Here we have the analysis questions from looking at the cheek cells. We used iodine to stain the cheek cells, so we'd be better able to see them. In the center of the cells, there was a dark structure that was stained a little bit darker. That tended to be the nucleus. And the cheek cell is an animal cell. It's not a plant cell, it's an animal cell. So now we're going to look at the onion cell. This is the onion through the microscope, shot with my cell phone at using the 4-power lens. You can see that these little boxes or little blocks are they're rectangular shaped, and that's because they are plant cells. Plant cells have a cell wall and a cell membrane. The membrane is difficult to see, but the walls are very easy to see. Those walls give them the structure and the rigidity that they need to reach for the light, for sunlight, so they can do photosynthesis. So this is the 40 power here. 10 of the eyepiece and 4 power objective. And then if we go to the next one, they get a little bit bigger and clearer. Again, shot through the microscope with my cell phone. Pointer, you see the boxes, the blocks of the onion cells. We have a nucleus here, another nucleus here, one there. They tend to be off to the side in plants. Again, boxy for structure and rigidity. And then finally, we have the 400 magnification. Really close in, we have a nucleus, we have the cell membrane, cell wall, another nucleus, probably a scratch underneath, another nucleus right here, and another nucleus right here. Maybe we can zoom in a little bit. And might even be able to see some of the chromosomes in there. Anyways, these are the onion cells. Those are the things that I would hope you would have seen. And then as far as the analysis questions for the onion is different from the cheek cells in their shape. And the onion is more rectangular because it has a cell wall that gives it structure and rigidity. And the onion is not an animal cell, it is a plant cell. So hoping that that this is some of the things that you were able to see. If you were unable to complete this and get all these tasks done, I am having a win time on Tuesday for students to come in and would given the opportunity to finish up the lab using the microscope. So hope you're having a great day. Talk to you soon. Take care. Bye-bye.